Today, an FDA approval in breast cancer, a review period extended in acute myeloid leukemia, promising findings in a urothelial carcinoma trial, a European approval in non-small cell lung cancer, and highlights from the 2019 AACR Annual Meeting. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has expanded the approval of palbociclib capsules in combination with endocrine therapy for male patients with hormone receptor positive, HER2-negative, advanced or metastatic breast cancer. The approval is based on real-world data from electronic health records, which demonstrated encouraging response rates with the CDK4/6 inhibitor in combination with an aromatase inhibitor or fulvestrant in this patient population. Results also show that the safety profile for men who received palbociclib is consistent with the tolerability in female patients who were treated with the agent. A detailed analysis of these data will be presented at an upcoming medical meeting. The electronic health records and post-marketing reports that the approval is based on are sourced from the EVIE Insurance, Flatiron Health Breast Cancer, and the Pfizer Global Safety databases. While some therapies are gender neutral in their indication, others have been indicated only for women, although they are often prescribed for male patients. Current clinical practice standards state that male patients with breast cancer are treated similarly to women with the disease. In acute myeloid leukemia, the FDA has added 3 months to the review period for a new drug application for quizartinib for the treatment of adult patients with relapsed refractory FLT3 ITD-positive disease. This will allow the agency to review additional data provided by the manufacturer Daiichi Sankyo. The application was filed based on data from the Phase 3 Quantum-R study, in which quizartinib reduced the risk of death by 24% versus salvage chemotherapy in patients with FLT3 ITD-positive relapsed refractory AML after frontline treatment with or without hematopoietic stem cell transplantation. Results showed that at a median follow-up of 23.5 months, the median OS was 6.2 months with quizartinib compared with 4.7 months with salvage chemotherapy. The OS benefit was demonstrated across 3 pre-specified sensitivity analyses: censoring for the effect of transplant, censoring for the use of other FLT3 inhibitors, and the per-protocol set of patients who were randomized and treated without significant protocol deviations. The survival benefit was also observed across several patient subgroups, such as those who had prior allogeneic HSCT and intermediate and unfavorable risk scores. The FDA initially granted a priority review designation to the NDA in November 2018, with an action date set for May 25, 2019. Under the new timeframe, the agency is now scheduled to make a final decision on the application by August 25, 2019. Topline results from the Phase 2 EVIE-201 trial showed that enfortumab vedotin showed strong clinical activity in patients with locally advanced or metastatic urothelial carcinoma who previously received both platinum-containing chemotherapy and immune checkpoint therapy. In the single-arm study, enfortumab vedotin induced a 44% objective response rate per independent review. The duration of response was comparable with findings from the Phase 1 EVIE-101 trial, which supported the breakthrough therapy designation for enfortumab vedotin the FDA granted in March 2018 for the treatment of patients with locally advanced or metastatic urothelial carcinoma who progress on a PD-1 or PD-L1 inhibitor. Results of EVIE-101 showed that among 71 evaluable patients across the entire cohort, the ORR was 41%, which included 3 complete responses and 26 partial responses. Sale Genetics and Astellas, the manufacturers of the agent, plan to present further findings from the trial at an upcoming medical meeting. Based on the EVIE-201 data, the companies also intend to submit a biologics license application to the FDA by the end of 2019. In non-small cell lung cancer, the European Commission approved dacomitinib for the frontline treatment of adult patients with locally advanced or metastatic disease with EGFR activating mutations. The approval is based on data from the Phase 3 Archer 1050 study, which showed that dacomitinib reduced the risk of disease progression or death by more than 40% and resulted in an average 6.5-month improvement in response duration versus gefitinib as a frontline treatment for patients with advanced EGFR-positive disease. Results also showed the median progression-free survival for patients who received dacomitinib was 14.7 months versus 9.2 months for participants who received gefitinib. The median duration of response was 14.8 months with dacomitinib versus 8.3 months with gefitinib. At a median follow-up of 31.1 months, additional data presented at the 2018 ASCO Annual Meeting showed that the median overall survival was 34.1 months in patients randomized to dacomitinib versus 26.8 months in those randomized to gefitinib. The FDA approved dacomitinib in September 2018 for the frontline treatment of patients with metastatic NSCLC with EGFR exon 19 deletion or exon 21 L858R substitution mutations based on the Archer 1050 findings. The 2019 AACR Annual Meeting took place in Atlanta this past week, highlighting therapeutic developments across a wide range of malignancies. For example, in neuroendocrine carcinoma, results of the Phase 2 DART trial demonstrated that the combination of nivolumab and ipilimumab induced a greater than 40% response rate and was well tolerated in patients with high-grade disease. Additionally, the 6-month progression-free survival rate and median overall survival with the immunotherapy combination were 31% and more than 11 months, respectively. Historically, these rates had been approximately 10% and 3 months, respectively. Findings of 2 expansion cohorts of the TATAN trial, which evaluated the combination of osimertinib and the MEK inhibitor savolitinib in patients with EGFR mutant MET-amplified non-small cell lung cancer who previously received EGFR TKIs, were also presented at this year's meeting. Results demonstrated encouraging clinical activity and an acceptable risk-benefit profile. In the first cohort, which comprised patients who progressed on first or second generation EGFR TKIs, the overall response rate was 52%. In the second cohort, which included patients who progressed on a third-generation EGFR TKI, the ORR was 25%. Finally, results of a small study presented at the conference showed that CAR T cells targeting mesothelin-expressing tumors demonstrated safety and efficacy in a preliminary clinical evaluation in patients with malignant pleural disease. Data showed that 13 of 21 patients had persistence of CAR T cells in peripheral blood from day 1 to 38 weeks, which was associated with a greater than 50% reduction in mesothelin-related peptide and evidence of tumor regression on imaging. Objective responses occurred in 8 of 11 patients who received a combination of cyclophosphamide conditioning therapy, the CAR T cells, and at least 3 doses of an anti-PD-1 agent. There were also no on-target off-tumor toxicity, no evidence of immunogenicity, and no severe toxicity. This week, we sat down with Dr. Dipti Gupta of the Memorial Sloan Kettering Cancer Center to discuss the cardioprotective role of androgens.
It is important to understand the role of androgens in cardiovascular health and well-being before dwelling into the pathophysiology of ADT and its effect on cardiovascular risk. So as it turns out that androgens have a significant cardioprotective effect. In the 1990s, there were a multitude of studies that looked at the role of androgens as coronary vasodilators. So if you injected intracoronary testosterone, it led to coronary vasodilatation. In men with established coronary artery disease, testosterone, when given, prolongs the time to ischemic ST depression on treadmill testing. Furthermore, testosterone helps with plaque stabilization by inhibiting platelet aggregation and promoting fibrinolysis and clot degradation. As we all know, testosterone also has favorable effects on body composition. It helps improve and increase lean body weight, meaning it helps to gain muscle mass, which also improves insulin sensitivity. Last but not the least, we know studies have shown that men have shorter QT interval as compared to women. In an NHANES study of greater than 700 participants, the higher the testosterone level, the shorter the QT interval. Now, even though we don't know the clinical ramifications of this finding, we do know that prolonged QT is antiarrhythmic. So we can speculate that shorter QT intervals probably lend antiarrhythmic properties. As we can almost assume, and as we already know, that there's a lot of shared risk factors between prostate cancer and cardiovascular disease by virtue of risk factors as well as age, as well as the demographic. So, and in addition to that, some of the therapies, including androgen deprivation that is used for prostate cancer, often tend to have cardiotoxic effects. We'll talk about that a little bit more in the future. But because of all those reasons, it is not surprising that cardiovascular mortality is a big cause of mortality in patients with prostate cancer. And most studies have shown that it trails prostate cancer mortality by only a little bit. So it's a significant amount of patients with prostate cancer that actually die of cardiovascular reasons, not of prostate cancer itself.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.